welcome and thank you for coming. So it's just a quick bio workshop. I think the email and that Google Forms would have sort of let you know what it's for. I know you all probably had a very unique year 12 and I am sorry for that. I know it's probably not the year that you guys were expecting, and having exams you pushed so far back is not ideal either, but hopefully there's some sort of benefit in that it's giving you some more time to, I guess, get it all together. Today, the structure is just at the start, we're going to go over just some tips and tricks for the exam, for basically any exam that's kind of like bio, that's a bit more content heavy, and then more specifically into the 2020 study design, things that I think will come up on the exam. So like a bit about me, I'm obviously not an examiner or anything, but I have been tutoring bio for 4 years now. I actually did my 3-4 bio when I was in year 11 in 2014. So the study design's definitely changed since then. But I still think the tips and tricks that were useful then are still very useful now. So yeah, if you have any questions, like I've been repeating, send them in the chat or feel free to unmute yourself at any time. If I'm not seeing any questions or anything, just unmute yourself and let me know. And yeah, I'll give you points at which there'll be points like when I'm referring to the study design or whatever that you can ask about specific like topics. So definitely use that if you feel the need to. So we're going to start with the 2020 study design adjustments. So the things that have been removed from it, from everything. So I think by now you're all probably doing practice exams, and you would have noticed that all of the exams like 2017, 18, 19 still have stuff about chromosomal abnormalities, things like aneuploidy, polyploidy. That stuff's been taken out, but things like block mutations, like inversions, deletions, and insertions, those are still in. So make sure that you can differentiate between the two, because you still need to know that there's a mutation when the chromosome's inverted or when something's gone or when something's added. You just don't need to know about aneuploidy, which is about the karyotypes, so things like Down syndrome, having abnormal numbers of chromosomes, things like that. Change over time, so divergent evolution, convergent evolution, mass extinction, that's been taken out. Mitochondrial DNA in terms of analysis, you do need to know what mitochondrial DNA is. Just to recap, mitochondrial DNA is obviously DNA in the mitochondria, and it's passed down by the maternal lineage. So you will have the same mitochondrial DNA as your biological mother, and everyone has the same mitochondrial DNA as their biological mother, and so it's actually conserved really heavily over time. Things like DNA hybridization, sequencing, and profiling have also been removed. Things like cultural evolution and like changes in geological history, rational drug design, and Relenza. So Relenza is an antiviral drug that's been taken out, but you can actually, like, there are some questions where they ask you to compare antiviral drugs to vaccinations, and obviously they can't ask you about that this year, but what they can do is just compare antibiotics to vaccinations, right? So antiviral drugs are very similar to antibiotics, except for the fact that like an antiviral drug is specific to a virus, whereas an antibiotic can cover a wide range of bacteria because it's sort of targeting their like plasma membrane, so it can affect more than one. Whereas antiviral is very specific to the unique antigens on different viruses. And yeah, so antibiotics you need to know, antivirals you don't. Yeah, so the point here is that none of these topics were ever large enough to warrant their own question, right? So it's not like from past exams you're cancelling a whole question 10 out. All you're cancelling are those smaller part C's or D's. But in saying that, majority of the stuff that's been cancelled is from Unit 4. So I do think that means there's going to be a much heavier focus on Unit 3. So having those early topics like cellular respiration, photosynthesis, diseases, enzymes, all of those, signal transduction, all of those down pat is really important. And like evolution is obviously a big topic, but you're only going to see like maybe 1 or 2 short answer, whereas the Unit 3 is going to be the bulk of it, especially for this year. I think there's a question. Okay, all good. Yeah, sorry. If you have a question, just send it in the chat. Okay. So these are the topics that I've already been mentioning that I want you to know inside out. With mitochondrial DNA, that's what the question was about. You do need to know what it is. You just don't need to know how they sequence it. In the same way, you don't need to know about how to DNA sequence. Like you don't need to know about how you're extracting mitochondrial DNA from the mitochondria, how you're sequencing it, and how you're comparing it. All you need to know is what it is. So it's lesser than you had to know before 2020, if that makes sense. So biomacromolecules, all of these topics, I don't want you to be studying them as individual topics. I don't want you to have notes for biomacromolecules and just tick it off. I want you to see how they relate to each other. So biomacromolecules are inherently related to enzymes, inherently related to antibodies, right? You'll talk about proteins, you'll talk about primary, secondary, tertiary, quaternary, and then that's going to come up in how what protein function is. So enzymes are big functions of proteins for globular proteins, and then you've got antibodies as well. You've also got plasma membrane proteins. So all of these things are related, and you're going to see Vcaa is going to relate them all in the short answer anyway. So you're better off, instead of having those very segmented topic by topic notes, I want you to start integrating things in your mind maps. I am conscious that there isn't long left till the exam, so you're probably not rewriting notes, but I think things like mind maps where you just have protein in the middle, and you start just linking every single thing you know about protein. What's a protein? You start talking about enzymes, and you start talking about how enzymes and substrates link. Then you start talking about antibodies. You talk about the specific nature. You talk about complementary shape. You talk about peptide hormones. Do things like that just so you can see how interrelated the entire bio study design is, even though it doesn't seem that way when it's initially taught to you, because those are the sort of aspects that Vcaa are looking for. And so I think it's really beneficial for you to start linking things together in that way. Proteins, I think, are a big one because protein synthesis is also massively tested for you guys as well. So things like transcription, translation, signal transduction, which obviously relates to that as well. Lac operon, I think knowing the lac operon is very important here as well. But yeah, these are obviously the big ones. In terms of human evolution, I think you don't need to know to a very deep extent about each species. I think, as we progress, they want you to know a bit more about Neanderthalensis and Denisovans and the crossover and the fact that there was coexistence there. But you don't need to know too much. So don't focus on the bio aspects that are just very rote learn based. Focus on the ones that are logical processes, because that's where I think the marks are. So going over the study design in general, I think a really important one that's often overlooked is that final area of study that is about experimental design. With a lot of the other topics in 3-4 bio, it's just about kind of knowing the content. They can ask you to extrapolate a bit further, but your answers are rooted in how much of the content you know, how much you can sort of remember, how much of the steps you understand. Whereas with experimental design, you're never going to get an experiment that you've seen before for the most part, right? They can ask you about anything as long as they give you enough background information. So it's all about you adapting your understanding of something and translating it to these different examples. And that's why I think this is an area where students often do poorly, and it's an area that's increasingly taking up more of the exam. What you're going to notice is, and we'll talk about this later, but where students do poorly is where they're going to keep assessing, right? And with science in general, bio 3-4 is trying to kind of get you ready for science or bio in uni, and that's increasingly become more research based. So having these key principles of research methods, having the basics of independent variables, dependent variables, understanding why experiments are set up the way they are, and understanding how to set up your own is going to be really beneficial, because I feel like they're going to keep trying to pull those those traits out of you. So going through this section of the study design is super important, because they're just concepts to know. There's nothing to rote learn. There's no definitions to rote learn. It's about understanding and being able to apply it. So I reckon that it's definitely worth your time. So this is sort of how to start, like these are sort of things that I want you to do in terms of studying. Practice exams and checkpoint questions over notes, 100%. What you'll notice is Vcaa, they're not creative. You're going to see either the same question, like multiple choice, exact same question, or very similar questions where they're changing like one little variable here or there from year to year. Right? So the more questions you're exposed to, the more likely you're going to have seen your exam before even actually seeing it for the first time. And I think, yeah, it's such an important thing to know. Even if you're like, oh, I haven't had enough time to go over Unit 4, it doesn't matter. Use those old Vcaa Unit 4 exams and just see where you need to concentrate your study, right? Practice exams are such a good way of highlighting which areas of the content that you're struggling with, because it's so, like, it's kind of easy to look at your notes and be like, okay, I know about evolution, but it's another thing to be able to apply that knowledge when they're sort of like critically assessing you. And it's, yeah, so it's like you can know something by like having a vague understanding, but there's a way that Vcaa wants you to know things, and it's that ability to kind of go a bit deeper. And you only realize how much you don't know when you've kind of put yourself under that pressure of doing exams and practice questions. So like definitely from here on forward, I don't think it's that important to be writing notes or perfecting notes or rewriting things. I think it's more about just doing as many questions as you can, especially for something like bio. In saying that, you need to be marking these questions. It's one thing to be doing them and exposing yourself to them, but it's another thing to actually be marking them and learning from your mistakes. But one of the best guides that you're given are those examiners' reports. Not only because they give you like the answers, but also they tell you, like, they give you some sort of insider info on what Vcaa are looking for. If they're giving you a 3-mark question, they're actually saying, okay, 1 mark of this is for this aspect, another mark of this is for this aspect. And I feel like that's something that's really important, and that's something that you should definitely look into. Be harsh with your marking. I know it's so, like, I know from trying to mark my own exams that it's like, you know, when you read something, you're like, oh, that's a bit off, but I knew what I meant. Just because you knew what you meant, only you know what you meant, right? A Vcaa examiner isn't going to know. Your friends aren't going to know. So maybe even with those questions where you're unsure, get someone else to read over them and see, did you get this answer from this? And if it's a no, get that mark off, right? Because then you'll read the examiner's report and you'll say, oh, this is a much more clearer, more concise way of saying what I wanted to say. And it's fine. Like, it's fine if you're getting marks off in your practice exams. Like, don't use that as a deterrent. Don't use that as, you know, like, I'm screwed sort of mentality. Look at that and be like, well, I tried my best. I've been exposing myself to all of these questions. And now that you know the correct way to answer them, you're not going to make that mistake again. Finally, looking over the examiner's report, one thing that, again, that I was talking about before that I've noticed is if you see that over 50% of students are getting 0 for that question or getting half of the marks for that question, they're going to ask questions like that again, or they're going to use that as a guide on what to focus on for the next year. So if you're looking through, for example, for the 2019 look, if you're looking through the multiple choice and marking it and you're seeing some questions where people are just not getting the marks, those are the areas that you need to focus on, because that's what Vcaa is going to try and get people to improve on, right? Because if they notice that this is an area that needs attention, they're not going to just like remove that from the study design. They're actually going to heighten it, and they're going to ask more questions about it, which is a bit cruel, but they're going to do that. And I think in their mind is beneficial for the student. So use that to your advantage. But yeah, so I'll send this out to you guys. So don't worry about like taking too many notes. Okay, so for short answer questions, quality over quantity. I know that time can be a pressure for some people, but honestly speaking, like, you know, 2.5 hours is a long time. There are a lot of questions, but you will have enough time to get through them all. Do not think of it as a race. Do not look at the person next to you and think they're already on this question, I need to speed up. Don't think of it as a race. Like, make sure you're taking that time for each question to think about where you could be improving. Make sure you're rereading some questions. Short answer is really tricky because sometimes you can feel pressured into writing more than you have to, and then when you write more than you have to, if you've added something that's wrong, you're actually losing marks there. Dot points. Dot points are your best friend. You do not need to be fluent, right? There are no marks for fluffing up your short answers. You don't need to keep massive sentences and keep adding things. All you need to do is be concise and to the point, and they will actually reward you for doing that, because the marking system is 1 mark for this keyword, 1 mark for this keyword, 1 mark for this key point. If you're sitting there writing paragraphs upon paragraphs, it's going to be hard for them to find those keywords and those key points, and they're not going to be reading those conjunctions and things like that either. So save yourself the time and save them the time as well, and just use the dot points. Be aware of the marks. So marks can inform you the number of points that you need to give. So those are some examples there where it's like sometimes if they're asking you for examples, right, and there's 2 marks, that could either mean 2 examples or that could mean 4 examples. It depends on what it is. If they're giving like an example of a time where, then it's probably 2. But if it's just like, oh, what are some controlled variables here, and it's 2 marks, then that could be 0.5 marks each. Be aware of like the question stems as well. So the marks can inform you of that. Like if they ask you to explain something, it's always about that definition. It's always about going a bit deeper. It's not just, okay, in this graph we see that the rate is decreasing, right? A lot of people say that because they're summarizing what's happening in the graph or in the table. But the explain or the describe why or the describe how is always about, this is the trend, this is happening because of this, right? And sometimes it's not clear why, but you need to extrapolate. You need to understand that, okay, the temperature's high, maybe it's outside of the optimum range for the enzyme, and so the rate has decreased. Things like that. I feel like you need to, you just need to know what the keywords are in the stems, and that comes with practice. Timing is everything. If you haven't already, you need to be doing these exams to time. It's more about that pressure that you're feeling that's important to help you across these practice exams. It's sort of about your self-awareness as well. You need to know, are you someone that's fast? There are some people that are just naturally really, really fast in exams, and they need to learn that practice of patience and going a bit slower. And there are some people that take their time, and then that means that they're rushing for their final questions. So then this kind of means you need to play to your strengths. If you're someone that's slow, then maybe you're better off doing your short answer first and then coming back to your multiple choice, where you might have less to think about, or you have, all the answers are there. So it's about crossing things out, and you're sort of like salvaging time there. And if you know you're going to be pressured for time, you're better off guessing like 3 multiple choice than having to put some random answer down for a short answer that you didn't get to read properly. So thinking about how you're going to feel in the exam, what that pressure of time, what that pressure of being in that exam room is going to make you do, and just being aware about playing to your strengths is important as well. So this is the 2019 examiner's report. This is like the paragraph before the actual answers. And it's fairly important to just go through this. So I've already highlighted the important parts. Here it says students were less confident in their identification of the types of errors that can occur. So this is within experiments and that like experimental design. So that's again one that I think is going to come up heavily in your exam. So for this one, it's all about learning experimental design, errors, limitations, confounding variables, and how to fix them. They're usually going to give you a question about what's an error that this student did and how could this be fixed, right? Common errors are like not having a control group, potentially if it's like anaerobic respiration, like are they keeping oxygen out? Those things are more specific to what they're testing, but not having a control group, having confounding variables, like potentially other things outside of the independent variables having an effect on the result, on the results shown, not repeating it enough, human error, right? If they're describing an experiment that students have done and they're saying that they didn't calibrate this thing properly, human error is another one that you can talk about, but that's probably the least priority one. You want to be talking about legitimate things like the experimental design first. Some questions require more thought and application. is important students that have prepared answers are very likely to earn them full marks for this type of response. So for things like natural selection and allopathic speciation, there can be a methodology that you sort of follow to answer the questions. You can have like little mnemonics and things to remind yourself of what do you need to, like what points do you need to bring up, like variation, gene flow, things like that. You have them in your head, but sometimes if you're not adapting that to the question style, if you're not using the species name or you're not using the specifics of the question, then it's going to look like you've just copied and pasted a response, because you sort of have, right? So it's good to go in there with devices that can help you, but you need to make it specific to the question itself, because Vcaa will see through it and they will not give you marks because of it. Okay, so the next one was, so about data and diagrams. A lot of these data and diagrams can be a bit tricky. So it's always about like those extended answers for after they've shown you a stem with a diagram. It's always about going a bit deeper. It's always those explain, describe how, describe why questions, where not only are you showing what's there, you're trying to add a reason why. And that reason why is never in the stem. It's always rooted in your theory. And that's why it's so important to know things back to front in this way. And I think like I just think students do poorly in this because not many people know some topics back to front. I don't actually think it's the fact that they didn't realize that explain meant to go a bit deeper. I just think that they didn't have, like, they don't have the knowledge there, or it's not reinforced as much during when like teachers are teaching it. But now that you know that, like, just look a bit deeper, especially into experimental design. Why do experiments with enzymes? Why do experiments with photosynthesis, cellular respiration, why do they go wrong? And increasingly experiments about like antibodies, monoclonal antibodies, things like that as well. Look into those. Yeah, and so a logical approach, clear answer, followed by the justification. So again, this is a marks type of thing. If they're giving you two, if they're asking like student A thought this, whereas student B thought this, who was right, you always need to compare. It's going to be a 2-mark answer, and that 2-mark is first for point 1 is this person is right because of this. Point 2 is this person is wrong, or therefore this person is wrong because of this. Not only are you proving someone right, you need to prove the other person wrong. It's always about the comparison. And again, those dot points and being more concise is always more beneficial. Like, do you agree with this? You can genuinely just have dot point 1. No, I don't agree. Or no, they're wrong. Why? They're wrong because blah, 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 blah. And I think it's so much better to clear it out that way. Or what's happening here? You label the thing and then you define it. Just help yourself be more clear. Yeah. Okay, so November 13th, so that's coming up. Remember, using reading time wisely, having a game plan, and playing to your strengths. So now we're probably going to head and do the 2019 exam. We're going to do the short answer. When I say we're going to do them, we're just going to go over what the questions are asking for, and for the more difficult questions, we'll actually answer them as well. I have a link for you all so you can sort of follow along. Give me a moment. Okay, so I'll put it in the chat for you. Okay, so let me know if you can all get onto this one and if it's working for you. And as I type, it should add to it as well. Cool, thank you. Okay. Sorry, just give it a second to load. Okay. So with bio, all of the short answer and the multiple choice are in chronological order. So your first questions are going to be the early topics of Unit 3. Usually half of the exams Unit 3, then Unit 4. So by question 6 or 7, you're going to have those Unit 4 questions. With 2020, I think it'll be sort of like a 65-35, Unit 3, Unit 4. And yeah, so play to your strengths. If you're better at Unit 4, start halfway through section, the short answer. If you're better at, you're better at Unit 3, then go chronologically. So this question, they're showing the 2 molecules. Hopefully you can identify molecule 1 as being tRNA and molecule 2 as, molecule 2 as being just your classic DNA. And then when they're asking for 2 differences, all they're asking for are the differences between RNA and DNA. And these are things that you would know from Unit 1 and 2, also from earlier this year. So the differences are so there's 3 differences between DNA and RNA. One is the nucleotide. So in RNA, they have A, C, G, U, so U for uracil, whereas in DNA, it's A, C, G, T, T for thymine. So you can just put in difference 1, uracil, thymine. And then the second one is that your tRNA is actually single-stranded. I know it doesn't necessarily look it, but it's single-stranded, whereas the DNA is double-stranded. Another difference you could put is that the tRNA would have a ribose sugar in each of the nucleotides, whilst the DNA has that deoxyribose sugar in each nucleotide. So that's fairly straightforward, 2 marks. Okay. So then they're giving you a section of 10 amino acids forming a protein. The section of RNA found in the nucleus of the cell associated with the translation of the 10 amino acids was found to contain over 300 monomers. So explaining how there can be 300 monomers, but only 10 amino acids. So firstly, they're talking about the actual RNA molecule. So that RNA molecule is mRNA, but we can assume that the RNA molecule that they're talking about is their pre-mRNA, right? Which means that the pre-mRNA contains nucleotides for introns as well as exons, and obviously the introns get spliced out, but that means that those are non-coding, right? So introns coding. Another thing that you need to mention is obviously there are 300 monomers, but for when you're talking about translation into an amino acid, 1 amino acid is coded for by 3 mRNA nucleotides, right? So even if it was with the 300 monomers, you divide by 3, you've got 100, right? So obviously you need to account for the 90 other codons that exist. It's possibly likely that the majority of those are introns. Also, I mean, technically here they don't show a stop codon, but if they did show a stop codon, then mRNA is long. mRNA, like transcription doesn't stop at when there's a stop codon, right? Transcription stops when a promoter of the next gene is present. Translation stops when there's a stop codon. So that means that a section of mRNA can have a bunch of exons that are coding for amino acids beyond a stop codon, but obviously translation's only going to stop there. So it doesn't apply for this question because they're not showing the stop codon, but for a question that did, that would also be a valid answer. Remember, if you have any questions, just put it in the chat. Okay, the next one, so we're on to photosynthesis. So a chloroplast is surrounded by a double membrane, 2 molecules as inputs for photosynthesis that would need to diffuse from the cytosol. So whenever they're asking for 2 molecules and there's inputs of photosynthesis, there's only 2 inputs of photosynthesis, so the rest of the information is not really as relevant. But diffusing from the cytosol also implies that they are either gas molecules or they're small uncharged, or it's water, right? So you know that the two inputs, or I hope you know the two inputs are water and carbon dioxide. Okay, so then it says, okay, so a higher concentration of oxygen is found in region R when a plant is photosynthesizing compared to when it's not photosynthesizing. Account for differences in oxygen concentrations found in this region. So, and region R is within the chloroplast itself. So it has nothing to do with the oxygen that would be being produced by cellular respiration, because I think some students in the past have said that. So this is all about why is there more oxygen in the stroma. Well, oxygen is produced by photosynthesis, right? So when photosynthesis is occurring, when the light in the light dependent water is photolyzed into O2, therefore oxygen needs to be, so in light dependent, oh sorry, my spelling, stage of photosynthesis, water is photolyzed into H plus ions, electrons, and oxygen. The water then, the oxygen, sorry, then diffuses into the stroma. Because remember, the light dependent stage actually occurs in the grana, but the oxygen leaves the product, so it diffuses into the stroma, and then it will eventually diffuse out of the chloroplast into the cell. So you need photosynthesis to be happening, where you need access to light for oxygen to be present within the stroma. Yeah, or you can like say region R. So basically, like that's kind of your one mark. The second mark is basically accounting for the difference, right? So when light is not available, water cannot be split, and therefore no oxygen will be produced. And no oxygen will be produced, therefore nothing can enter the stroma. Okay, describe the role played by each of the coenzymes NADPH and ATP in photosynthesis. So it seems slightly complicated. It's not. Like, obviously you don't need to know the biochemical pathway of NADPH or ATP. You just need to know about, in relation to that Calvin cycle, what's going on. So all you're seeing is NADPH and ATP are being recycled, right? So once they're charged, they're going into the light independent stage, they're going into the Calvin cycle. NADPH will transfer hydrogen ions, and then ATP will transfer energy. And that is all you need to say. You don't need to be more specific beyond that. I know it seems like you have to, but that's all you need to say. It's 2 marks. They've given you 2 molecules or 2 coenzymes. So all you need to do is 1 mark for what each one of them does. Another thing I want to mention is that Vcaa will always, unless it's like a 1 mark question, they will always give you more lines than you need, and do not be fooled by trying to fill those lines up. I've seen so many students that will try and fill all 4 of these lines up, just like, just for the sake of it, or just because they feel like they have to in order to get the 2 marks. You most definitely don't. And because it honestly isn't enough for you to say for a question like this to fill the 4 marks up, you're going to end up saying something that's wrong, just to compensate for the lack of space that you've taken up, and then you're going to lose the mark, right? So you're actually kind of, you're sabotaging yourself by doing that. So they're going to play mind games with you. They're going to give you more lines than you need to, but don't fall for the trick. Okay. The human immune system consists of a series of defensive barriers that protect the body from infection. When bacteria come into contact with the body, they immediately encounter these defenses and must bypass each barrier if they are to survive and infect the body. When bacteria come into contact with the body, they must gain access to the living tissues to become pathogens. List 2 possible routes the bacteria could use to access living tissues of the body. So whenever you're seeing possible routes, they're basically talking about the first line. So it's like the first line of defense, and these are your mechanical chemical barriers. First line of defense, so they're also like the processes or the systems. That wasn't meant to be in caps. So basically all of the systems, a good one, so if you're thinking like your mouth, that's your digestive system, right? Mouth and nose, that's your respiratory system. If you think of like COVID, respiratory system, reproductive system. And then like Vcaa, for this question, they've also just accepted a cut in the skin or eyes, right? So obviously those two aren't systems, so they're not necessarily roots, but they're still places where infection can begin and where infection can enter the body. So this is what I mean by roots is just a vague way of saying first line of defense. They're just areas that once a pathogen can get through, that's entered the body. And that's basically any part of the body that's like compromised, any part of the body that like there's a cut or any sort of like entrance for infection, that's a first line of defense that's been broken. Okay, so once bacteria are within or have access to living tissue in the body, but before cells are aware of their presence, bacteria will encounter chemical barriers. List one of the chemical barriers and explain its function. So you would have gone over this in the first line of defense again, where they talk about mechanical barriers and chemical barriers. So mechanical barriers are things like your skin, things like skin is probably your main mechanical barrier, teeth, things like that. Chemical barriers are, like we say, chemicals, so they're more enzyme based. So if you think about like lysozymes in like tears on your eyes, even like mucous membranes, that's kind of, I mean, that's a mix between chemical and mechanical, because it's a physical barrier, but it's made of chemicals, so it's up to you under what you place that in. A good one that I always put for chemical barriers would be stomach acid. So inside your stomach, you have a very low acidic pH, and that's to help digest proteins and things like that. But it also can kill bacteria, right? Bacteria are unicellular organisms. They have enzymes. They have proteins to make up their outer membranes, and so if they fall into acid or a pH that's outside of the optimal range for those enzymes, then they will denature, and then that bacterial cell will die. So a good one that I would say is stomach acid. There's a very low pH which can be outside the optimal enzyme. Sorry, I'm typing kind of fast. Okay. Yeah, other things that you can say, again, lysozymes in tears, those are just digestive enzymes, so bacteria sort of like trying to get in through tear ducts. The lysozymes will hopefully take them out. Yeah, so that's a good one. Another one that Vcaa has listed is complement proteins. So complement proteins, they haven't specified that it has to be the first line, right? That's why you can mention complement, because complement's part of the second line and it's bacteria specific. I would probably stay away from mentioning it, just because when I look at this question, I think about first line of defense. But if they mentioned any sort of innate defense against bacteria, complement is a really good one, because it forms like a membrane attack complex and it kills bacteria directly. Another thing that Vcaa has noted was that for students that talked about lysozymes, some of them said lysosomes. So lysosomes are an organelle inside cells that contain lysozymes, which are digestive enzymes. But obviously in tears or on the surface of your eyes, you don't have lysosomes. You don't have random organelles. What you do have are the enzymes. So spelling, like I said, in bio, English fluency and spelling doesn't matter. But if you spell something wrong and that actually means something else. So if you spell lysozymes differently and it actually reads as if you were trying to write lysosomes, you can lose a mark. So just be careful. It's like if you accidentally write ADP instead of ATP, you could get that mark off because adenosine diphosphate is different to triphosphate, and they'll give you the mark off even if it was like an accident on your end, right? So just be wary of those sorts of things. When an inflammatory response starts, the first cellular responders will be cells from the innate immune system. One of these cells releases histamines. How does histamine contribute to the inflammatory response? So histamine should be something that you know from more about, like from the allergy responses rather than like the immune or like pathogen response. Basically what histamine does is that it upregulates inflammation. One of the main things it does is that it increases permeability of like blood vessels, so it causes them to dilate. It's something that we call vasodilation. And then this allows more white blood cells, so macrophages, neutrophils, to the area of infection, and they will upregulate inflammation by releasing cytokines. So what histamine does is histamine increases vasodilation, which enables more macrophages. There's a bunch of white blood cells that you would have gone through in Unit 3, but just all of them, like macrophages, neutrophils, dendritic cells, all of them are attracted to the area to come to the area of infection. Okay. If bacteria are not destroyed by innate immune responses, adaptive immune responses become involved. Describe how an adaptive immune response is initiated during a bacterial infection. So this is basically how does the innate immune system connect to or join with the adaptive immune system. And I think a lot of you would know this answer already. It is your antigen presentation, right? Your antigen presenting cells, dendritic cells, and macrophages are a part of the innate immune system. They will display anything that's foreign. It doesn't matter. It's not specific. There's not like a specific type of dendritic cell for a specific type of pathogen. They will engulf anything and everything. In saying that, they would only engulf things that are non-self if they're working correctly, and then they will display their antigen in place of their own MHC marker. So APC is short for antigen presenting cells. I don't want you to use acronyms in your answers, but I'm just going to do it for the sake of time. And they will engulf non-self cells or pathogens and display the non-self antigens in place of their MHC markers and display this to T helper cells. Antigen presenting cells, a good thing to remember is that they use the lymphatic system to move. So they're like this is helpful for time because moving through the circulatory system takes a long period of time, but the lymphatic system also connects the lymph nodes together. So you'll usually have them engulf something at the site of infection, and they'll use the lymphatic system to make their way up to the lymph nodes, which are about here. And that's where they'll interact with the T helper cells. And then from there, that's your adaptive immune response. The T helper cells will either stimulate the humoral or the cell-mediated or both, depending on what it recognizes the pathogen to be. Okay. So this question is about thunderstorm asthma. The things that I've highlighted are things that I want you to highlight ideally. I think it's important. Yeah. I think it's important to highlight during the exam. I really think it's such an advantage. Obviously not the entire question, but just keywords like this. Like when you see allergy, like the word allergy, you already know histamines, mast cells, like those words should already be coming to a head. If you study using that mind map technique, like that's a really good way to condition yourself to see a word and have some sort of recall. I don't know if any of you do psych, but if you do, they'll teach you all these little learning techniques about recall. So for this one, hay fever and asthma. Asthma is obviously its own condition and it has its own things associated with it, but hay fever is just your basic allergy response. So pollen fragments can cause allergic reactions when they enter the body by interacting with a specific type of cell and protein of the immune system. So another keyword that I did not highlight here is interacting. I know when we think allergies, we think mast cells and we think release histamines. Histamines are little proteins, but remember that the pollen, which is the allergen, isn't actually interacting with histamines. That's an indirect interaction, right? So what I want you to note there is that this question is asking about things that the pollen or the allergen will directly interact with. And histamine is something that it indirectly interacts with. So that's not a correct answer here. So the specific type of cell is your mast cell. And the protein is the IgE or the immunoglobulin E antibody present on mast cell surfaces. Right, so here the 2 marks are for the answering each part of the question, the type of cell and the protein. But knowing that immunoglobulins are just antibodies and all antibodies are proteins, IgE antibodies are specific to allergy. So if you just said antibodies, that wouldn't be good enough, or just immunoglobulin, that wouldn't be good enough, because IgG and IgM are more about different types of pathogens like bacteria and like virus infected cells, whereas IgE is specific to allergens. Okay, list 2 strategies that a person could take to reduce their risk of developing thunderstorm asthma. So this sort of question is going to be similar to a type of question that you guys could get on pandemics, right? They could easily give you like, oh, this X is a pandemic or an epidemic in your area. What is something that someone could do to reduce their risk? Sort of a similar deal here. Asthma and like hay fever, they're respiratory diseases as well, because pollen is coming through the respiratory system for the most part. So what you would do there is wearing a mask is one that works for both the pandemic situation and for allergies. Another good one is antihistamines. So these are things that you would know from actual experience, or things that are just sort of common sense. These aren't things that you're learning. These are just things that you just know. And then also like just avoid going outside in high pollen environments. Right, so these sorts of things, very self-explanatory. Some students struggle for these ones, like even in the pandemic questions, because I feel like you look at these questions and you always think like it has to be harder than it is, or you think that there's some sort of trick here. There isn't. Sometimes Vcaa gets lazy and they're giving you a free-for-all question. Take it, take these marks and run. Same thing with the pandemic situation. Like if they're gonna give you a free 3 marks to give, like, you know, potential solutions or practical ways to limit exposure, you know, and you've lived through a pandemic this year, so use those things that you've acquired and put them in, like they're free marks for you. Okay, explain if it could be possible to develop a vaccine against thunderstorm asthma. So this is an interesting question, and this is the question that a lot of people got wrong. It's 2 marks, 54% of students got 0. So what you sort of need to realize is allergens are different to pathogens, right? Pathogens come at the detriment to a host. They're going to cause disease, whereas allergens are things that are mistakenly identified as being like non-self or dangerous to the body. So with pollen, pollen does induce this sort of reaction with people who have hay fever, but there are so many different types of pollens. Pollens are different for each plant, right? So if you're technically looking at antigens on the surface of pollen, each pollen is different. You can't actually find, there's no common antigen for all different types of pollen because it's all different for all these different species of flowers. So basically you need to say that this is not possible. Vaccine against allergens or pollen here, because you're not allergic to one pollen, or in the same way that you get an adverse reaction to one bacteria, right? It's the combination. It's, you can go outside in winter and you can be okay when there's like one pollen or like there's a low pollen count, but the minute spring comes and you've got a very high pollen count, that's when the allergies hit, and it's because of that compounding effect. And so you're not going to be able to find a vaccine that accounts for every single species of pollen, and B, acknowledges that compounding effect. What would actually happen is it would actually induce the immune system to have a more adverse reaction, and there would actually be like inflammation by cytokines and things like that as well. So this would actually cause more harm than it would good. So what you need to know is a vaccine would result specific species of pollen, which would in turn upregulate and especially for things like pollen, because you can't control your exposure to pollen in the way that like a food allergy, for example, if you're allergic to shellfish, you can you can obviously not eat shellfish, but you can also potentially take some sort of vaccine against like little additives in food so that you can prevent yourself from being from feeling as sick as usual. But with things like pollen, you're going to go outside, there's going to be pollen. Like there's no way to prevent that unless you're like legitimately living in a bubble. So there's no way to like create a vaccine, because you would have this massive upregulation and there would be actual cytokines released, and that would cause a lot more harm to your airways and potentially result in the airways constricting and closing at a much faster rate than someone who just had a normal hay fever reaction. Okay, so this question is interesting. It's sort of like testing the remainder of the genetic component that you got left in Unit 4, and it's sort of about evolution as well. They mentioned BMP4 a little. I think the majority of it has been taken off the study design, the same way that like Darwin's findings and the Galapagos Islands have also been taken off. For these questions, you don't actually need to know anything about BMP4 because it's all in the STEM. So you could technically still answer this one. Basically, I've highlighted that BMP4 affects beak depth and CAM affects beak length. So if you see BMP4 and CAM highly expressed, you look here, you see, all you need to do is where is BMP4 high, where is CAM high, and then you see where BMP4 is high, it's deep, high, it's long, and so then your answer should be deep and long. So don't spend too long on these. Like whenever there is a table or a figure of some sort, the questions are pretty easy. They just try and make it look confusing. Okay. Another gene found in birds, the gremlin gene inhibits the expression of BMP4 in the cells of the feet. Using your knowledge and information above, explain what would happen to beak shape, depth, and length if the gremlin gene also had an effect on the cells of the beak. So here you get 2 marks. The 2 marks are for talking about what happens to beak shape in terms of the 2 parameters tested, so depth and length. So your first point would be if BMP4 is inhibited in the beak, then the beak would be short, right? Because low or absence of BMP4 or low BMP4 has a shallow beak, right? Because only high has deep. So the beak would be shallow, as BMP4 has no effect on this BMP4 expression on CAM expression, then there will be no effect or no change in the length of the beak. So, yeah, self-explanatory, but yeah, like sometimes the question seems deeper than it is. It's not. It only relates to one parameter, and the other one you can just say there's no change. Okay. Explain why a mutation that affects the expression of BMP4 can lead to rapid changes in the phenotypes of the Galapagos finches. So this one I am a little confused on in terms of whether it's like still relevant to you guys or not. I think this is less relevant than the rest of the questions they've asked, because potentially it requires on some external knowledge of BMP4. The answer here is that BMP4 is a master gene. Master gene means that it has an effect on the expression of many traits. So it controls, like it's not about the genotype, but it's the fact that when BMP4 is expressed, it can highly influence the phenotype of many different traits, not just like beak depth. But also, like they mentioned, in feet, like all of these other traits that you can see in birds, right? So when BMP4 is altered, or when the expression of BMP4 is altered, that also results in the altering of phenotypes, and that means that it changes like the expression of these like a multiple range of phenotypes, and that means that there are selective advantages and disadvantages that are newly shown, and that could result in, I mean, that's just natural selection, so then that has different traits that are then going to be selected for, and that could change the whole like makeup of what traits are selected for in this entire species, resulting in the entire species to be changed. So with this one, I would say that it's not as relevant for you guys. So I would say like 1 mark of this is relevant, and I wouldn't leave it at 3 marks. But I'm just going to explain that for you, but it's not something that you necessarily need to know. Okay. Question 6. So from 10 weeks into pregnancy, okay, from 10 weeks into pregnancy, a woman can decide to have a non-invasive prenatal test that affects fetal aneuploidies. A blood sample is taken from the woman for the test. So what does aneuploidy refer to? So that's off your study design for 2020. I'll just let you know, an aneuploidy is an abnormal number of chromosomes, so either too many, so above 46 or below 46. But again, it's off the study design. What would scientists examine in the woman's blood sample? This is one of those, whenever you see these like random 1 mark questions, they're just like logic-based questions. You're not bringing any theory to the table here. You're just talking about like, you're basically just using the STEM to help you out. If they're taking blood and it's a prenatal test and it's about fetal aneuploidy, they're taking fetal DNA or fetal sample. So the sample contain fetal DNA. So this is the test that they would do at the end of the first trimester, just to see if things are okay. And they would obviously need to test the fetal DNA and not the maternal DNA, because you know that the maternal DNA is fine. Okay. If there is a requirement to look for other genetic abnormalities, such as block mutations, different procedures can be conducted. Describe 2 different types of block mutations. So they've gotten rid of aneuploidy and polyploidy in chromosomes, but they didn't get rid of block mutations. So these are the ones that I mentioned at the start. So insertion, deletion, inversion, translocation. So insertion is when addition of segment of chromosome, addition, or I guess removal or deletion. So that's the same. Insertion and deletion are very self-explanatory. Inversion is when you swap. So inversion is when you like segment of chromosome swap. And then translocation, so trans is like different chromosomes. Location is when two different chromosomes swap genetic information. So inversion is on that same chromosome, the top and the bottom switch, whereas translocation is when from like chromosome 23 to chromosome 12, top and bottom swap. And that's usually more dangerous. So segment. And I think we have a question potentially. Okay, you should have access to the document. I can send it again. Okay. Some procedures can detect single gene mutations. Sickle cell anemia is produced by a single gene mutation that affects the production of the beta chain of the hemoglobin protein. Beta hemoglobin is a single chain consisting of 147 amino acids. Shown below is a small section of the mRNA nucleotide sequence found in an individual without the mutation. So this is what would technically be happening normally. Use the section of mRNA on 24 and the codon table above to complete the table below. These codon tables are fairly self-explanatory, so I'll just go through it with you. Whenever you see AUG, that's your start codon. You should know that that is methionine. Or you can obviously look at this, A, U, and then G, so you're at MET. Then the next one is GUG, so GUG, that's a vowel. CAC, CAC, that's his. CUG, CUG, that should be leucine, ACU, threonine, CCU, proline, GAG, glutamine, and GAG again is glutamine again. So that should be right. Okay, the gene for sickle cell anemia produces misshapen red blood cells that do not carry oxygen efficiently. If an individual with sickle cell anemia, the 20th nucleotide has changed to uracil in the transcribed mRNA on page 24. So just count by 3, 3, 6, 9, 12, 15, 18. This one has become a U. Right, so it's GUG. So if you're looking for GUG, that's val. So how will this alter the amino acids coded for this sequence? Well, the glutamine, or you can just say the glue, whatever, in the seventh position, mRNA would become a valine. Or would be replaced by a valine. If the 20th nucleotide was deleted, how could this mutation alter the structure and function of the hemoglobin protein produced? So you'll notice that these two questions are obviously different. This is just about how would this alter the amino acids in the sequence. So just talking about how what happens to the old amino acid, glutamine, becomes this new amino acid. This one is about the structure and function. So they're not actually asking you to be like, oh, the glutamine becomes XYZ. They just want you to tell them what's going to happen to the protein now. So if now instead of the U, we just cancel this out, then what happens is from here you get GGG, and then you get AG, whatever comes next, right? So then that means that's a whole frame shift mutation now, because your glutamine has now changed to GGG, which is glycine, and now it's going to change the next amino acid, which is going to change the next amino acid, and it's sort of like a domino effect. So if 20th nucleotide was deleted, this would result in a frame shift mutation. So frame shift, the entire frame has to shift one forward, and then that changes every single amino acid, which changes every single amino acid forward, and basically, and would most likely, because if every single amino acid from there on was affected, that basically results in some in a non-functional protein, right? Or it's definitely not going to be hemoglobin as you know it. Having a point mutation is obviously not ideal, but it doesn't change the entire protein. Clearly there are issues that become sickle cell anemia, but it's still recognizable as a hemoglobin, even though it's faulty. If the whole or a whole segment of the amino acids are different, that's basically either non-functional or a completely different thing, probably a disease-causing agent. So non-functional. Yep. But yes, it will most likely become non-functional protein. If you want to address the structure, so the function would be is that it's non-functional now, and then the structure you would say as the shape is irreversibly. Okay. So this question 7, we've sort of moved into the classic Unit 4 stuff, going into evolution. So you're seeing a rock hyrax, elephant, dugong, and then these 3 species are closely related, sharing recently common ancestor, but have different features, right? What's the type of evolution? So these questions are actually easy. This one's off your study design. So convergent evolution, divergent evolution, and mass extinction are off the study design. For reference, this would have been divergent evolution because of the recent common ancestor, but that's fine. The next question is that in 2014, paleontologists discovered a frozen, well-preserved, complete specimen of an extinct species, the strep bison in Siberia. This intact specimen was dated at 9,300 years old. Scientists are debating the cause of the strep bison's extinction. Give one possible cause of its extinction. So, the reason I've highlighted the 1 mark is, again, it's one of those classic 1 markers that are nothing to do with the theory that you have and all to do with common sense. You can really say anything here. One thing that comes to mind is that when they say frozen, well preserved, you could just genuinely say ice age, right? You know, there's been a couple of ice ages in the Earth's history, so an ice age is a possibility. You can talk about the cold environment, which, like, instead of an avalanche, or the cold environment resulting in a lack of sustainable environments, habitats for these animals, habitat destruction, disease, lack of genetic diversity. But yeah, so like even if you just said ice age, like 2 words, that will get you the mark. Okay. So this is a 4 marker, so watch out for this one. So they're asking about, describe what would have occurred to lead to the preservation of the animal from when the strep bison died to when the frozen, well-preserved remains were discovered. So this is basically your steps of fossilization. If it's asking from when the animal died to now, you need to have like a logical sequence set. So from when it died, the first thing that you would have learnt about fossilization is that there needs to be some sort of rapid burial. In this case, you can say rapid burial, or because you know that there's a very cold, snowy environment, you could just say that it was frozen. Like it died and then it was like encapsulated in ice or something like that. So you need to mention rapid burial or frozen, like, I don't know, immediately frozen or like covered in ice, which is sort of similar to rapid burial, but you need to mention something like that. So that's like, and you can expand on this, you can make the sentences sound nicer, but these are this is the crux of the information that you need to put in. So rapid burial, frozen, covered in ice. And then you need 3 of these points. You basically need to talk about like they would have given you a list of things that you need to know in terms of what is the correct environment or the correct conditions of fossilization. So rapid burial is like the first thing that needs to happen, and then from there onwards there needs to be a lack of scavengers, or like a lack of scavenge, like the body can't obviously be eaten. It needs to not be able to decay. So if it's buried under a bunch of rocks, or even if it's like frozen, it needs to be in an environment where it doesn't decay, right? So this means very warm environments are sort of out of the question here, which is why the cold environment is ideal. It can't decay there. There needs to be a relative lack of oxygen, because oxidization leads to decaying. And there needs to be some sort of like erosion, melting or something to expose the remains. So you need to come back in that circle of what happens as it dies and what happens to enable it to be discovered. So it's rapidly buried. Then you'll talk about body is frozen, so it doesn't decay. As body is frozen, scavengers would not have access to it. And then over time, ice begins to melt. You can add in a cheeky nod to climate change there. The glaciers start to melt, and then the remains begin to be exposed, right? Ice begins to melt, tides rise, whatever, and the remains become exposed, allowing them to be discovered. So you need to address the things that they're asking for, right? They want to tell you, they want a point about when it died, they want a point about when it remained, and then the two in the middle are up to you. Okay, so the Genomics Health Futures mission will run a $32 million trial starting in 2019 to screen over 10,000 couples who are in early pregnancy or who are planning to have a baby. Using a blood test, individuals will be screened for 500 severe, deadly recessive genetic mutations. Couples will be told they have a genetic mutation if both individuals in the couple carry the same mutation. The trial may lead to a population-wide career screening program. The research will evaluate cost effectiveness and psychological impact, ethics and barriers to screening. It is anticipated that future tests will be free of charge. So give an example of a disorder or disease that can be detected by genetic screening. Okay, so this can be anything. I know in your study design, genetics isn't a big part of it. It needs to obviously be a disease that is passed down genetically, and not an environmental one, but anything you can think of, anything that you've heard about, Huntington's, cystic fibrosis, Down syndrome, anything that is rooted in genetics, but exclusively rooted in genetics. So I'll just put like Huntington's, but anything, you can put anything there. Again, those classic 1 markers. Blood sample from the individual will provide researchers with only a small amount of DNA, so they're going to use PCR to amplify it. Okay, so the 3 stages of PCR. So whenever we talk about genetic manipulation, manipulation techniques, obviously recombinant plasmids and gene cloning is a big one. Then it's PCR, then it's electrophoresis. As we mentioned at the start, so much of these techniques have been taken out of your study design. So those 3 are the ones that you need to know back to front. As you see, like PCR is one that you need to know the stages of. So I will quickly go over them. Stage 1 is denaturation. So your temperature is at 95 degrees, and then high temp results in hydrogen bonds, DNA strands being broken, DNA becomes single-stranded, right? Then your next step is called annealing. So then temp lowered to 55 degrees, primers attach to DNA, Yeah, primers attached to DNA. That's really all there is to annealing. And then there's elongation. Elongation is an interesting step because then the temp is increased to 72 degrees. And then Taq DNA polymerase adds DNA nucleotides and forms new DNA fragments. Right, and then this process is like repeated a bunch of times. So, and then DNA doubles each time, right? So you go from 2 to 4 to 8 to all the way up to however many you need. Elongation is interesting because the temperature's increased, but it's not increased to 95, right? The point is increasing temperature increases kinetic energy. The more kinetic energy, the faster the reaction rate. The faster the reaction rate, the more, I guess, the faster the reaction can occur and the more DNA that's going to be produced, right? So you want to have this balance between efficiency and denaturation. So obviously 95 is too high, but 55 is too low. So 72 is a good in between. The other thing to notice and another thing that you need to put in is Taq DNA polymerase. DNA polymerase alone will not cut it because our DNA polymerase, obviously the optimal temperature is like 37.5 or 40 degrees and it will denature quickly from there on, right? Taq refers to a thermophile bacteria. Thermophile means it loves heat. It lives in like those little, in like volcanoes basically. And it has, therefore, because it lives in these very high heat environments, all of its enzymes have very high heat optimal temperatures. And so we extract those and we use them for like lab purposes. And that's because they can obviously handle very high temperatures and allow a very fast reaction or very efficient reaction rate. So make sure you put Taq in front of DNA polymerase. Okay, discuss 3 factors that affect the migration of DNA fragments through the agarose gel. So this is obviously talking about gel electrophoresis. And it's basically asking you to type down every single factor you know about it. So one of the main things that you should know is that DNA is negatively charged. So it will be attracted to the positive end of the well and move towards it. The next one that you should know is, so DNA is negatively charged, and then the second one is it's about the size, right? All gel electrophoresis is separating DNA molecules by size. So the smaller the size of the DNA, the further it will move down the agarose gel. And the last one, I mean, this is like less stress. These first 2 points are things that you probably already know. The last one is just you sort of need to think about it. What powers the like gel electrophoresis is actually a voltmeter. You've got a voltage current being sent through, and that's what powers these things to move further down, and that's what also gives it the poles, like the positive negative. Another one is that like agarose is a gel, right? If you think about even like agar gel, like the agar gel that you put bacteria on to grow on. I don't know if you ever got to do those experiments for your area of study 3 in unit 3, but you usually get to put bacteria on an agar plate, and that's like thick gel is the same gel that electrophoresis is done on. And so like the more dense the gel, the slower all the molecules. And similarly, talking about the voltage, the longer the voltage is applied, the further the DNA will move, right? Yes, of course, the shorter ones are going to move furthest, but if you like allow this to go for 24 hours, those DNA fragments are going to move off the gel and they're going to fall off. So the length of the time that voltage is applied is correlated to okay. Again, if you guys have questions, just shout out or put it in the chat. Okay. The test may find that the couple who are planning to have a baby or who are already pregnant both carry the same severe or deadly mutation. Describe one ethical and one social issue or implication that could arise from this finding. So again, these questions are sort of like BS type questions. Just you reading the, like you're not bringing anything else into it, you're just reading the stem and you're trying to answer it. Ethical and social issues, technically both of them, there are a lot of issues that fall under both ethical and social. I guess ethically you can talk about the fact that you can talk about like quality of life. All of these are a little dicey in terms of if you're like in this situation where they're pregnant and they want to keep the child and they find that the child is carrying a deadly mutation, it's like for them ethically they're wondering, is it worth going through with a pregnancy if they know that it's going to result in death anyway? So there's things like that. And then obviously there's like questions of abortion. So like, you know, is it like something to do in terms of should we terminate the fetus now? Should a severely ill child be born? Like what's the quality of life there? These are all things that you could mention as ethical issues. So I will leave that up to you there. Social, I guess, is again more about the society as like a wider institution. So if you think about like the cost of these sorts of testing, the cost of like IVF and things like that, psychological implications. So like on a more social side, the fact that like now these parents have to go away with it, like knowing the fact that they're carrying this deadly mutation, they might not be able to have like healthy children, they might not be able to have children. So there's like psychological impacts there for them and also other parents who are finding this sort of news. And you know, like from a society side of things, like is there support offered, right? From the doctor's clinic where they find out this information, are they referred to like psychological services that can sort of help them mitigate these issues? So those are things that you can answer. Again, it's not very bio, but it's up to you what you want to answer here. There's no direct right answer. You just want to make sure that if you're putting something down that's ethical, you want it to be something that's ethical, so something that's kind of tough to answer. There's no right answer here. It's just up to this individual, and it's a tough decision. With social, you want it to relate a bit more to the wider context of things. Okay. So question 9 is about Zika virus. So you can read the stem. Part A is about that Zika fever is thought to occur when pathogen infects humans from an animal host. Identify one social or economic factor that could lead to transfer between hosts. So social, economic, a lot of these, so there are a bunch of these ones. You could say that like potentially those who are poorer or have a lower socioeconomic status live on outskirts of like suburban areas or outskirts of metropolitan areas, which means that they border areas that have a higher that are like more, you know, animal concentrated. So if they're saying from monkeys, for example, it's unlikely that these animals are going to be in the heart of the city, right? And so if you think about socioeconomically, people who are living in the city potentially have more money, people living in the outskirts have less money. Therefore, the people who are living further out are actually more likely to get into contact with these animals because they're living closer to the border of the animal's habitat. Right. Another thing you could say is that economically, as population's increasing, we're moving further to the outskirts, right? We're building more, we're cutting down more land. And so the animals, something that was once the animal's habitat is now being destroyed. And these animals aren't like, they don't know what's going on in that way. And so they're going to start, there's going to be a period of time where there's overlap, right? As humans keep growing where we're living, these animals are going to be coexisting with us, and that's going to enable human to animal transfer of diseases. So you can talk about that from an economic standpoint, or the first point is more about like how socioeconomic factors put people on outskirts. So there are a couple things up to you. Again, this is kind of a BS question, so it's kind of up to you how you want to answer that one. So when scientists attempt to identify a particular disease, they look for specific antibodies in infected humans. Scientists trying to identify Zika virus infections found that the testing for the antibodies produced against the Zika virus often gave them incorrect results. This was because the antibody tests that had been developed could not always identify the difference between the antibodies produced against the Zika virus and antibodies produced against other viruses. So explain why making a correct identification of a viral pathogen is important in control of the disease. So this question's actually fairly hefty. It's got 3 marks for it. So there are it is obviously very important to correctly identify the viral pathogen. Different pathogens have different modes of transfer, different modes of transfer, like in terms of how they pass from one person to another, different modes of transferring within the body, what cells they're attracted to within the body, like in terms of infecting, which cells they're going after, also about like specific antigens on specific cells. So like if you're looking to make an antiviral drug, knowing the specific pathogen is really important, because you don't know what they're like, there's like a capsid protein that encapsulate most viruses. All of them have different antigens, so knowing which one it is would be very helpful in being able to produce some sort of antiviral drug as well. So different pathogens, or you can add like viral, because that's what the question asks for, have different modes of transfer from from infected individuals. That's about like so that means different ways to protect the community. So like, for example, COVID is respiratory, but viruses such as like malaria and things like that, blood-borne viruses, those are obviously different, right? You're looking out for different things. There's different ways to protect the community, and there's different campaigns that you have to do for these viruses that are transferred in different ways. Then also different viruses affect the body differently and obviously target different cells or different pathways, and that's important as well. So important to know for symptom to like deal with symptoms, right? They're going to go after different things. Also to know to be able to diagnose. Like COVID is super vague, right? Like there's, some people can't smell, some people are coughing, some people have a fever, but not everyone has the same thing. Whereas some other viruses, everyone's like vomiting or everyone's getting diarrhea. Like those sorts of viruses, at least when people have it, they know they have it. The issue with COVID is, compared to those other viruses, you don't know that you have it. So the final one is different viruses, different capsid proteins, so different antigens. Obviously write these out as sentences, don't be lazy like me. Different antigens and therefore different antiviral drugs. Right? Because as we mentioned at the start, antiviral drugs are not like antibiotics. You don't have broad spectrum antiviral drugs because antiviral drugs target specific antigens, whereas antibiotics are targeting like LPS, like the LPS membrane, right? And like all gram positive or all gram negative have specific types of membranes, and so that's why you can have antibiotics that target more than one bacteria, but not antiviral drugs that target more than one virus. Okay, explain why the antibody test could not identify the difference between the antibodies produced against the Zika virus and antibodies produced against other viruses. In your response, refer to the structure of the antibody. So here 1 mark is answering the question. The second mark is adding the structure to your justification of your answer, right? So here what I would say is, if you can't tell the difference in antibodies, it's probably because the antibodies are similar. They have a similar shape. If they have a similar shape, that means the antigens on Zika virus and the other virus must be similar as well. So what I would say is the shape, antigen similar, and therefore the antigen binding site of the antibody must be similar. Or something like that. In that, I mean, that way I've just answered it in the one sentence. You can break it up into two, and you can say the antigen, the shape of the Zika virus antigen and the other virus antigen must be similar. And then you can say the virus will bind to the antigen binding site of the antibody. But either way, like this is enough to answer the question and get the 2 marks. Okay. Now, this is your classic pandemic question. Although Zika is just endemic, it's not a pandemic, but describe 3 different approaches other than vaccination that government health officials could use to reduce the spread of Zika virus, right? So for these ones, you can use the generic responses that you're all going to use if they ask about COVID, and then you can use ones that are like more relevant to the STEM, right? They've given you 3 marks, so you've got 3 approaches to talk about. So the generic one could just be like quarantining people, so like quarantine of people who come from like areas where Zika is endemic. Right, so they use a mosquito vector, right? So they're not found in every continent, they cannot fly great distances. So with mosquitoes, it's about like getting rid of the mosquitoes, right? The virus cannot spread without these mosquitoes, so you can control the population of the mosquitoes. What you can do is you can look for breeding grounds for mosquitoes, like still bodies of water, those sorts of things, getting rid of them, using repellent, using nets, those sorts of stuff. So using nets, fumigation as well, those sorts of things, like in the home to get rid of mosquitoes. And then another one that's always good is an education campaign. That's required almost everywhere for any possible disease ever. Education campaign. So this is like suitable clothing, like PPE, for example, how to protect yourself and what to do if you got it. Those sorts of things are obviously very important. You've definitely seen with COVID how important it is for the community to be educated, and how much fear, like, obviously comes as a result of a disease like this, of any sort of pandemic or epidemic. So education is super important. So you don't see the picture, so don't worry about the information in the article. But they're saying, would it be possible to classify Denisovans as a separate group from Neanderthals without using DNA analysis? So I actually looked up the site to see if I could get this. I can't. We're locked out. So knowing that we're not going to have all the information to answer this, I think it's just important to see that especially with species that are so closely related, like the early humans, DNA analysis is a must. It is a must to be able to differentiate, because we know there's overlap in terms of when some of these species were alive. We know that they've obviously evolved from a recent common ancestor, and so features, like facial features, like skull features alone aren't enough. It's not like some of these were bipedal, and like Denisovans were bipedal and the Neanderthals weren't. No, we all had those similar characteristics, so you can't really tell from the remains whether it was like this species or this species, which is why it's really difficult until you get to that DNA analysis to tell whether these skulls and these remains were from which type of early human. So for this, I would always be inclined to say no. I would just say, what the answer says is that it would not be possible to classify the Denisovans as a separate group from the Neanderthals, as the fossil sample is too small. Obviously, we don't know what the fossil sample is because we can't see it. Another thing that they've mentioned is, as both groups are extinct, you're unable to determine if fertile offspring could be produced. So basically, like, what they're saying there is, obviously, if you're the same species, two of the same species can mate to produce viable fertile offspring, right? And it's like a characteristic of being different species that you obviously can't do that. But like what Vcaa is obviously pointing out is because they're extinct, that basic experiment can't be done either. So for this, I will just say no. We wouldn't get the 2 marks without the information anyway, but just say no because these species are too closely related to be able to accurately from like remains alone, right? You need that DNA confirmation, especially because like we haven't seen many fossils from these different types of species, and like, you know, the Denisovans, we're only really finding out about them fairly recently, and so we don't have enough to be like, oh, like that would be a 20-year-old, that would be like, you don't have that knowledge, right? So when you're seeing a big skeleton, a small skeleton, you don't know if the big skeleton is because that's a Neanderthal and this is Denisovan, or because this is like a 40-year-old and this is a 10-year-old. Like, you just don't know. So with any question like this, especially for evolution, DNA analysis is a must. Scientists could have extracted mitochondrial DNA from the young girl's finger bone sample, but instead extracted and sequenced nuclear DNA. Explain why scientists chose to sequence the nuclear DNA for this sample. So, like we discussed earlier, mitochondrial DNA is heavily conserved, and you're getting it from the maternal lineage. So technically you have the same mitochondrial DNA as your great-great-great-great-great-grandma. And so that means that over time there has been little change to mitochondrial DNA, and it's probably fairly similar to the mitochondrial DNA of other early human species. And so it's much better off to use the nuclear DNA because that firstly is the whole genome, and it's more indicative of the genetic traits that are present in, like, that are currently present, right? Whereas the mitochondrial DNA, because it's not changing, it's actually more indicative of genetic traits that are from the past. So, so as mitochondrial mtDNA is inherited maternally, it is highly conserved and very similar, and could be early human or early hominin species. Nuclear DNA contains the whole genome and is more indicative of present day traits. Okay, question C, I'm sorry, we just can't do it without this because it's like very dependent on this diagram that's been copyrighted out. So we'll just skip that for now. Okay, so we're on to the last question. Question 11, you'll notice in a lot of the new ones are all about experimental design, and I think they're so worthy of paying attention to because they will definitely ask you questions about this, and they'll stress this in your exam. So this question is about investigating the activity of lipase. And so student 1 made the following notes before designing her experiment. Lipase catalyzes the breakdown of fats into glycerol and fatty acids. Fatty acids can neutralize an alkaline solution. Sodium carbonate is an example of an alkaline solution. So student 1 set up 4 tubes containing 5 ml of fatty acid solution, 10 ml of sodium carbonate solution. Each test tube was suspended in a water bath. Each water bath was set at a different temperature, and pH probes were placed into the test tube. Each solution had a pH of 8. After setting up the 4 test tubes in water baths, student 1 waited until the solution of the test was the same temperature as the water in the water bath. Student 1 then added 5 milliliters of lipase solution to each test tube. Student 1 timed how long it took for the pH of the solution to become neutral, so 7. Student 1 repeated the experiment so that the 3 measurements for each, so that she had 3 measurements for each temperature. She obtained the data shown in table 1. Student 1 was concerned about the precision of her measurements. Analyze the data above and state the temperature at which the measurements are the most precise. Justify your response. So for this question, you need to know what precision means, right? And precision is about, like, this is not a great definition, but precision is about having the closest or having the least difference between repeated experiments. So the least difference between repetitions of the same experiment. Right, because it shows that even if you're doing something wrong each time, at least then you're still doing the same thing wrong each time. So it's still indicative of the same experiment. So it's not like you're being precise because you're doing the same thing all 3 times or all 10 times that you're repeating it. And that's showing that that's shown by the fact that your results are very similar. So then analyze the data above and state the temperature where the measurements are most precise. So if you're looking through here, you're seeing 240, 238, 241. There's a bit of variation. Same thing with the next one, same thing with the next one. But here, 180 is actually repeated. And then this one is, I guess, a bit off, but not as much as the rest of them. So I would say at temperature 35 degrees, as there is the least variation between the measurement or the repeated measurements. Okay. State whether student 1 obtained qualitative or quantitative data. Outline a difference between the 2 types of data. So your 2 marks are for the 2 questions they've asked. So student 1, the data that they've obtained, the data that they've obtained are these measurements, right? The time taken in seconds. So that's a number. By virtue of it being a number, it's quantitative. Qualitative is, as it sounds, about the quality, so it's more descriptive. It's about saying if she just wrote fast, or if it was like the color of the solution, like when lipids, like when fats when they're emulsified in the digestive system, they can change color. And if you're looking at that change in color, that's sort of a qualitative data. But this one is definitely quantitative. So student 1 obtained and that's your first dot point. Outline a difference between the 2 types of data. So then you're saying that or you can say like as it is numerical, and then you could say it is not qualitative, or like qualitative data is more descriptive, whilst quantitative, my spelling, is numerical. Okay. Student 2 repeated the experiment designed by student 1 the next day and obtained the data shown in table 2. Identify which of his measurements have errors. Give 2 examples of sources of errors for these measurements. So, if you look through the measurements, these look, I guess, fairly uniform. This one screams out to me because it's a lot lower. So does this one. And then 35 is like perfect again. So then I would say, identify which of these measurements have errors. That's 1 mark. And then the 2 examples are the other 2 marks. So I would say measurement 2 at 15 degrees and measurement 3 at 25 degrees have errors. Right, and then the sources for error, these ones are interesting. This is like, they didn't tell you anything about what student 2 did, right? They're saying that they're repeating the experiment, but they're not saying like, yeah, they repeated it well. So all you have to do is look back here and see where could they have screwed up, right? Anywhere where they could have screwed up. So what you're noticing here is that measurement 2 is lower and measurement 3 is lower. So they're both lower than they should be. So what would cause the time to be lower? So if we're looking at the time taken for the solution to become neutral, so the time where, 8 to 7. So if the time taken for it to become more acidic or for the pH to become neutral, that means if it's going faster, that means there could either be more enzymes. There could be an issue with the calibration of the pH meter, like if he messed up calibrating the pH meter and it accidentally showed pH 7 earlier than it should have. Sodium carbonate is basic, so if he didn't add enough sodium carbonate, that would make this the solution look more acidic, so more like closer to 7 than 8. Or just like, yeah, incorrectly measuring the pH is sort of the same thing. So I would go like that. And then one of the last things that I would go for is like recording the time incorrectly. That's still a legitimate thing to say. Like, you know, he measured it, it was actually 2:30, but he looked at it and he just thought it was 1:55. Like, that's something that you can say, but it is a weak one, given that you've got all of this stuff to work with, right? All you want to make sure is that the errors that you're putting down are consistent with the fact that the measurement is lower than it should be. Because if you're saying stuff like, oh, added too much sodium bicarbonate, which is alkaline, that would make the solution more basic, and that would make it take a lot longer to reach a pH 7, right? So you want to make sure that it makes sense given the context. But I would say adding too little sodium bicarb, adding too much lipase, because obviously that would make the reaction go faster, incorrectly calibrating pH meter, which is the same as incorrectly reading pH, and then just incorrectly timing these experiments. They only want 2, so I would go with the first 2 if I was you, but these other 2 are still options as well. Later that day, the third student performed the experiment. Student 3's data is shown in table 3. After comparing his results with the other student's results, student 3 realized he had made an error. Name the type of error that was made by student 3 and identify one source of error that may account for the results obtained by the student. So, let me see. So it's meant to go down in general, but obviously this has gone too far down. So with this one, all of the measurements, so what you need to do is compare them to the other 2, right? The trend is correct. Like at 5 degrees, it takes longer. 35 degrees, it takes the least amount of time. So you go, the trend is correct, but all measurements are too low, right? So then that shows you that it's sort of like systematic in that every single, it's not like for, you know, measurement 2 of 15 degrees, he accidentally did this, or he accidentally did this for a one-off. It's every single thing is wrong. So that's telling you that there's actually something probably wrong with the equipment that he's using. The thing that this is called a systematic error. This is because it's not really human error. It's not a one-off, it's a system error because it's actually to do with the technology he's using. So the type of error is systematic. Sorry about the caps. This is something that you just sort of need to know the name for. And then one source of error, think about the systems that are being used that he's using. So it could be using like a wrong, like the timer that he's using. So timer that they're using be broken, pH meter could be incorrectly calibrated. Yeah, I think Vcaa, they also say the students are consistently using a measuring cylinder incorrectly, right? So every single time they're using this measuring cylinder, it's wrong. So I feel like I don't like that last answer as much, because if it's happening each time, human error is unlikely for you to mess reading something every single time. I feel like there has to be something wrong with the pH meter, because it's like every single time without fail, these measurements are too low. So I would go with the first two. You only need one of them, so one of the first two before I go with the last one. But if they asked you for 3 options, then you have to use that one. Identify a limitation of the experiment that was carried out by all 3 students. What could students do to address this limitation? So again, limitations for experiments is a big one, and I think it's one that students do struggle with. With this one, I think a big issue is there doesn't seem to be a control at all. There doesn't seem to be one where, because they're technically testing the effect of lipase as well. So they should have a group that doesn't have the lipase, right? They can do the same, like set up 8 test tubes instead of the 4, and for all of them you would have all those different temperatures, but you would just not have lipase in the tube. And just to see, like, is the change in the result exclusively due to the enzyme activity of lipase at the different temperatures, or is something else going on as well? So lack of a control group. I think this is a big one. All good experiments need a control group, right? So students add another 4 test tubes exposed to the same 4 conditions, except without the addition of lipase. That's the answer that I would go with. Another thing that you could talk about is the fact that so they're looking at a human enzyme, right? Well, lipase is something that exists within us, and they've only gone up to 35 degrees. We know that our body temperature is 37.5, so it is worthy going up to 40, potentially even going up to 50 to show what happens at denaturation. I feel like 5 to 35 is too low of a range for an enzyme that works in humans, and so it would have been a better and a more accurate experiment to have actually looked at a wider range of experimental temperatures. But that is it. So if any of you have any questions, now's your time to ask them. You can also email them to me. I've got some time if you want to ask any questions now. Put them in the chat if you don't want to unmute yourself. But yeah, up to you. Okay, cool. So I'm going to end it there. I'm going to put my email in the chat, and you all have it. I would have sent you an email last week anyway. So if you have any questions, email me. If you want any like one-on-one tutoring sessions, also email me. But other than that, I hope this was helpful. I mentioned the Healing Foundation at the start. It's an Indigenous-owned organisation that focuses on mental health and healing and trauma alleviation, especially for people that have, been affected by the stolen generations. And it's something that I'm really passionate about. So if that's something that you want to donate to, please go ahead. But otherwise, good luck for your exams. Thank you for coming, and you're gonna all kill it. So yeah, let me know how you go. But yeah, so I'm just going to leave you guys here.